tenebroso.
Está lá embaixo.
Exatamente. Sim. Muito mal. A sala é lindíssima, mas a coreografia é do pior que há. Em termos de intimidar o candidato.
É horrível.
Deve ser do José Belo, não?
O José Belo é do primeiro ano do programa, sobre o segundo.
Pois, está bem. Porque o ano em que eu lá fui, o José Belo também estava. Pois, mas talvez estivesse já no segundo ano da coisa, não sei.
Ou então depois desta era 2 anos.
Talvez 2 anos. Nessa altura que falei. A coisa sobre genética populacional, creio. Com o Luciano Terrenato.
Dos animais geneticamente modificados.
Também já fiz, agora faço linhas celulares geneticamente modificadas. Já fiz ratinhos.
Ratinhos geneticamente modificados. Qual era o que é que tinham de peculiar?
Neste caso eram genes envolvidos na formação das células que fazem os anticorpos, as células B. Não fazem nada a ver com comportamento. Esses fenótipos mais interessantes, realmente, vocês gostam mais.
Realmente a arquitetura genética do fenótipo é uma coisa fascinante. Por exemplo, o Hornless tem um gene, mas o espermatogene tem 2.000 genes. É simples.
2.000.
Há 2.000 genes que já se associaram à produção de espermatozoides, por exemplo. Portanto, é facílimo ver um homem infértil, porque há tantos pontos onde a coisa pode falhar, e realmente, até por razões ambientais, a qualidade de espermatozoides dos europeus está a piorar hoje. A baixa natalidade, entre outras coisas, também se deverá à grande infertilidade masculina. Outro fenótipo que eu agora na área da agricultura não sei nada.
Hello, good morning everyone. Thank you for joining us today in the first debate ever in the Bioengineering Week. I am Mafalda and I am the team leader of the scientific department for the VIII edition. Here you have the QR code for today's session. Don't forget to scan it with your camera so you earn biocoins to win one of our great prizes at the end of the week. Let me also remind you to submit all your questions through the SBE app, or you can just raise your hand and ask your question. So thank you, Professor Margarida, for moderating this debate, and thank you to our speakers. Without further ado, I hand the floor to Professor Margarida. Thank you.
So first of all, good morning, everybody. And I will probably try to skip to Portuguese, and I will immediately ask people in the audience if there is anyone here that does not understand Portuguese. Okay, so let's proceed with the English. No problem at all. This was thought since the beginning that the debate was supposed to be in English. So let's proceed in English. So first of all, thank you very much for the invitation to be here for the organizers of this debate, which is actually very interesting and very exciting. And first of all, I would like also to acknowledge and thank very much to the 3 participants today that were invited for this debate. Just some brief notes about their biography. So we will have, and starting with the lady, Dr. Helena Freire de Melo, thank you very much for coming, is a person from law. of course, that this topic of genome editing of the human genome, of course, has a number of ethical issues behind, a number of law issues. So, of course, we had to have here someone from law. So she has a graduation and a master in law. And interestingly, she's also the president of an association, VIII, that is dedicated to research, teaching, and dissemination of bioethics. Moreover, she's also the vice president of the Portuguese Bioethics Association. So she's a person from bioethics. Then we will also have Dr. João Lavinha. Thank you very much for coming as well. He has been during many years head of the Research and Development Unit, Human Genetics Department at the National Institute of Health, Ricardo Jorge. Everybody knows Dr. João Lavinha. And he has been, during all his life, a researcher, laboratory researcher. But in addition to that, he has also given a lot of reviews on ethics about biomedical research. So he's also a very interesting person to have here because of also this particular experience. Then we will also have Dr. Vasco Barreto. Thank you very much. He's an assistant professor at Nova School of Science and Technology, and he's an everyday researcher in the lab working with the DNA editing, genome editing. He has the DNA Breaks Lab, Gene Editing Services, a company called Gene Tatus. Okay, so after this this brief introduction, of course, I still don't have questions from the audience, but I think that for everybody here to be in the same page, I think it's important to give here some initial definitions, if you agree. So it would be important to understand what is genome editing in order for us, in the end of the debate, to understand if this is a very promising or dreadful thing as the name of this debate. So I would put this question to the three members of the panel. How can we define genome editing? In what contexts it can be used? And of course, afterwards, we will, after this part of these definitions, we will try to focus on the ethical parts, the security parts. So please, who wants to start? Some brief initial definitions of what is this topic that we will be discussing today. To start with.
Yes, Vasco is working on it, I think.
Yes, in the bench every day. So
I can give it a try as a biologist. First of all, thank you, Falda, for the invitation, and I extend my thank you to all the other organizers. It's always a pleasure to talk about these issues. Genome editing is essentially a phenomenon by which you change genetic information that we inherited from our progenitors. And that process can be natural or artificial. I guess in the context of this talk, we will be focusing on the artificial gene editing, but please keep in mind that many species have mechanisms of changing their germline DNA that they inherited from their progenitors throughout their somatic life. That is happening as we speak inside our bodies. We are editing the genes that form the antigen receptors of B and T lymphocytes. That's a process of gene editing that involves recombination of DNA material, mutations that may or not improve the affinity of the antibodies towards antigens, and that's important for our immune system. So keep in mind that gene editing is also a natural process. With respect to the artificial gene editing, I think we should establish two broad categories, which probably will also guide our debate. One is the gene editing of somatic cells, and the other is the gene editing of the germline. And the ethical questions that these two types of procedures pose are completely different. In the case of the somatic editing there are not that many bioethical issues. Most of the questions that remain are essentially technical, whether we can do it sufficiently with sufficient efficiency, whether we can make it affordable so that everyone can have access to these technologies that for the time being are still quite expensive. But there is no major bioethical question. In contrast, with respect to the editing of the germline, here there are a number of bioethical questions that we are faced with, and I think our debate will be revolving around these questions most of the time, I would suspect.
Okay, do you want to please complement the answer from Vasco?
Well, I think that Vasco has said the bottom line of the topic. The only thing that I could call your attention to is the possibility of using gene editing for not for therapeutic purposes, but for enhancement of, say, crop, wheat. You know that there are people intolerant to gluten. Wheat produce as this protein, the protein that people are intolerant to. So why not producing a wheat with less or no gluten in it? This is a good idea, and people have tried to do it, and apparently with relatively good results recently. The problem is, and this is just a model of a plant, so far away from us, although we will eat this wheat, and people used to say that we are what we eat. Nevertheless, the problem with this phenotype of gluten production in wheat is controlled by 35 genes. So you have to do not just one editing event, but you have to multiplex this approach. And the probability of having cross edition is exponentially increased with this multiplexing. And if we think of even the simplest phenotypes in humans or in mammals, like the one that I was commenting with my panel colleagues, spermatogenesis, which for each male is just a very basic achievement, is controlled by up to 2,000 genes. So bear in mind that if you wish to change a complex phenotype, you have to, it is, at least with the current technology, completely out of reach. So, and this is still a promise that the possibility of enhancement is still a promise in some literature, and there are meetings, and so. So, to me, this is a real hype. It's not something that we can envisage in a foreseeable future. So I think we should restrict to correcting very simple phenotypes, like monogenic rare disorders. The best example is an hemoglobin abnormality, hemoglobin S, caused by hemoglobin S, the sickle cell disease, which is in the forefront of the gene editing in humans, somatic, as Vasco was putting it. And now the patient organizations or the families of these patients, because they are mostly children, so the families are urging the regulators to free germline gene edition, which to my mind is far too early. And I would like to just to quote Jennifer exactly for the CRISPR, the gene editing procedure. She was saying recently in London last week in the third summit on gene editing, which took place in London last week in the Crick Institute, a moratorium is needed while we debate the ethics. So even the person who has devoted her life for gene editing is urging scientists to think back, scientists and the society as a whole, because it is not a choice of scientists, because scientists are kind of neutral regarding these things.
They only have to solve the technical issues.
Yes, but then the destinies of this technology are the people, are all of us. And not just the ones that have been born, but the coming generations. And if we manipulate germline, we are changing the human genome, the genome of the human species. And the descendants of these people that come from embryos that have been edited will inherit the edited traits. So the two restrictions to my mind, therapeutic purposes and somatic intervention. But of course, I am open to be convinced of the opposite.
Helena, do you want to compliment?
Well, thank you. I would like to say thank you to Mafalda for the invitation and to the other members of the committee. And what I can say is that we do not define anything in the sense that we accept the definitions that you in genetics and biology give us. And it's not an easy task in the sense that law comes after you have a scientific progress. The scientific progress is not good or bad in the sense that you are solving an issue that you decided to study, but the way you are going to use it may have very serious consequences in the society that we are going to have. Nowadays, you are even changing what we are as a species. And so what happens is that in the first moment you have a scientific progress, in the second moment you have a bioethical dilemma. Should we use the scientific progress or not? How are we going to use it? And then you have law. Law is always the last moment of this.
Regulates.
Yes, in the sense that it is a very heavy system. I don't know if you as students have the feeling, but from the moment you wake up until the moment you go to bed, law is everywhere. In what you can do, what you cannot do, how you should behave inside this room, if you can smoke, if you cannot smoke, if you have to sit in a certain order. So law and the sanctions that we have sanctions like prison, we have sanctions like fines, so have serious consequences in the way people live. So what happens is that law sometimes takes a little bit too long in order to decide how we are going to regulate what you are doing in your labs. And what happens is that many times when we enact a law, you are already doing something different. And we can speak about the leadership that when the United Kingdom enacted the law, the technique that they had in mind was not a technique that you used for reproductive cloning with mammals. So it is a very difficult approach, the legal approach, in the sense that we have the right to benefit from scientific progress. And it is a very important right in the sense that our life is much better having in mind what you're doing in your labs. Probably we would be dead if you did not do what you do in your day-to-day lives. But at the same time, what you are doing can change completely our society, and we have to decide if we want to change our society in that direction or not. And that has to be done within the democratic ways that we have to do it. And it's not easy, because usually what happens is that half the society wants stem cell research, half the society does not want stem cell research. Half the society finds out that the genetic modified organisms that you're creating are quite astonishing and amazing. Half of the society says, no, this is dangerous. If you release them into the environment and they and they procreate with other animals, what is going to happen? And then we are working with open concepts. Our main concept in law, in international law and in national law, is the respect for human dignity, dignity of the human person. We do not know what a human person is, but we all agree that it is important to respect the dignity of each one of us. And then we have different solutions within the same concept. You can be for euthanasia, saying that it is that it is admitted by human dignity, and you can be against euthanasia using the concept of human dignity. You can be for epigenetics using human dignity, and you can be against epigenetics using human dignity. So everything is open for us, everything is discussed for us, and at the same time it's very difficult because science is very, very, very fast. You are changing everything every day, and we are very, very, very slow. And we have to find a way of living between science, bioethics, and law, and it's not being easy in our societies. And also you have to consider that the solutions are different according to your preconceptions. And if you have a preconception that is linked to the religion, if you have a preconception that is linked to the political ideology, you will have a different solution in a way that you are regulating. And even if you do not have a preconception, but you have the same values in a society that tends to have a neutral approach that does not exist to these problems, you have different ways of thinking what is liberty, what is freedom. And so for us, everything is open, everything is under discussion. And what we tend to do is not to enact laws that are very strict, but to define general principles, general values, and try to not disturb you too much, but at the same time not have a society where you have a person that is half fish, half cow, half human, and that wants to involve here in technique.
Okay. So still in this attempt to put everybody in the same page and with the same initial knowledge about this, I don't know if Vasco or anyone of our member of the panel can tell us a little bit about the timeline of these technologies. I mean, since when it has been possible to do this type of human genome editing, and where are we now, technically speaking, in terms, for example, of efficiency? Because you talked about problems of efficiency. Mm-hmm. So when did all this start for the first time, and where are we now in terms of timeline?
Okay. I promise that I'm going to answer your question, but first I'd like to make a few comments. And and my suspicion was actually true. I think we have a problem here in this debate because we all agree on the main questions. So in order to create a debate, I'm going to adopt a contrarian position, which may not necessarily be mine, okay? Just to make it more interesting.
More exciting.
Okay? So I'll be the one defending the use of gene editing for human enhancement, which is something that I'm sure Elena and João Lavinia are really against, okay? Now, going back to what you were asking, so we started manipulating not necessarily the human genome, but I guess the main landmark in terms of technology applied to reproduction was in the 80s when in vitro fertilization appeared. So we had our first. At the time, we called it Bebé Proveta, right? It was in 1980.
Baby.
Right, yeah, that's it, baby, right. It was in 1986, if I'm not mistaken. And that was, at the time, it was really revolutionary, and it triggered a lot of discussion in society on whether we should be doing it, whether this is allowed. I mean, there were very strong religious views against it. But now looking back, I mean, this procedure is completely normalized. It's no longer an issue. There are still some important ethical questions related to in vitro fertilization, namely whether the person that is born out of this procedure is entitled to know the origin of the DNA in case a third party is used to provide the sperm cell or the egg.
Can I ask? Mm-hmm. Don't forget what you are about to say. Religiously speaking, this is an honest question because I really don't know. What does the Catholic Church, which is prevalent in Portugal, thinks about in vitro fertilization? Because for you and also for me, it's something that is quite established. I don't know the answer for this because, for example, in terms of stem cell research, Catholic religion is against the
they are against any type of manipulation, right? Any type of manipulation of the embryo. But as you know, I mean, the Catholic Church has been losing power quite dramatically, I would say, over the last 3 decades. So I don't think they are the main force these days shaping the debate. My main point here is that when it appeared, in vitro fertilization raised a number of concerns which are no longer valid. And the two main bioethical questions that remain is basically the use of surrogate mothers and whether the child that results from this procedure is entitled to know the origin of the DNA that she or he was given. But the procedure itself is completely integrated in society. Then in the 90s, you had essentially two main landmarks: gene therapy using very crude procedures, namely viruses that were modified to carry genes that were then inserted into the cells of the patients that had some defective gene. This was named gene therapy, and it worked, but there were a few cases in which it was a tragedy, either because the person developed a strong immune reaction against the vector, or because the vector got integrated in a region of the genome where it activated a proto-oncogene, and these people developed leukemia. This happened, for instance, in a number of children in France. And these two accidents, in a way, delayed the field tremendously. It was a huge trauma for everyone. At the same time, we had the birth of Dolly, the sheep, right, out of cloning procedure. So the scientists in Scotland were able to get a somatic cell from a sheep, to extract the nucleus, put it in a oocyte, and out of that oocyte, a new chip was born, Dolly. And this was in 1997, and it basically shaped the zeitgeist for the following years. And then you had movies with cloned humans. You had novels, for instance, who is probably today the most well-known French novelist, Michel Houellebecq. He published a book in 2005 called La Possibilité du Nil, which is basically about human cloning. Our José Saramago published in 2002 O Homem Duplicado, which was probably also influenced by these ideas. And there were a number of bad science fiction movies about super soldiers that were clones, right? And these days, we don't really talk about human cloning. It's no longer an issue, not even for science fiction. I mean, we clone dogs and we clone cattle, but this is no longer a main problem. But at the time, it was a major issue. bioethicists were discussing it, we were discussing it, law people were discussing it. These days, it doesn't seem to be a main concern. So the main concern these days are the designer babies. So the possibility of editing the germline to not only to cure monogenic diseases, on that we will all agree, but this possibility of enhancing our physical and intellectual and even moral capacities by changing the genome. And this became a question in 2013 when a nowadays very famous technology, CRISPR technology, João already mentioned it. So Jennifer Doudna was one of the inventors of this technology. She got the Nobel Prize in Chemistry a few years ago. And since 2013, we had this revolution in the gene editing field, an explosion of papers using this technology. And it's basically a technology that makes editing much simpler. So many labs that before were not allowed to work in this field because of a number of barriers now can do it because it's no longer as expensive as it used to be. It's quite efficient, and this basically led to an explosion of studies, and the progress has been outstanding. So now you have many different types of editing procedures, and the field keeps evolving at a very fast pace. There are many companies very well funded working using these technologies, mostly related to the cure of somatic diseases, because as you know, germline editing is forbidden in most countries, and probably it will remain forbidden for a number of years because it's not really a problem of efficiency. The technique seems to be quite efficient, and it has been improving. Although, yeah, that can still be a problem. I agree, but the main problem
the repair
is
system of the embryo doesn't work as well as the somatic repair system.
It's true, but I'm sure the efficiency will be solved as a problem. I mean, at the pace of evolution that we are seeing, we will solve that problem. What seems to be harder to tackle are the off targets. So the off targets are basically these additions, these mutations that occur outside of the region that we want to edit. Say that you want to edit gene A and put a mutation that reverts the mutation that was creating your mutant protein. You can do that, but at the same time, you will introduce a few mutations elsewhere in the genome, and you don't really control for this very well. And this may create problems. So although you solve the problem that you had initially, you created another one. So this is one of the problems that people are facing when it comes to the cure of monogenic diseases. But I should say that for most monogenic diseases, in fact, we don't need CRISPR, we don't need gene editing, because if you are going to edit the genome, then you are going to do an in vitro fertilization necessarily. You have to edit the egg in vitro or ex vivo. And if you are doing that, probably you are going to create multiple embryos. And by creating multiple embryos, you increase the chances of getting at least one embryo that has one good allele that is not mutated. Because it's very rare that the two progenitors will have all the alleles mutated. This can happen, but it's going to be extremely, extremely rare. So for most cases, you don't really need to edit the genome. You just need to do a genetic screening of these embryos and select the one that carries at least one healthy copy of the gene in the case of recessive diseases. In the case of dominant diseases, you will have to go for an embryo with two intact copies. But in many cases, this is also possible because the two progenitors will be probably heterozygous for the disease, even if the two are sick, which is again is unlikely. So I think it's a kind of a false problem, this idea of applying gene editing to cure monogenic diseases, because with the pre-implantation genetic screening, you can get rid of most of the monogenic diseases. So it's mostly when it comes to human enhancement that this technique is at the same time tempting and worrisome, right? And Joy is right. One of the problems of this technique is that for most of the traits that we are interested in, say intelligence or height or the ability not to become fat, you don't have one or two genes involved. You have dozens, hundreds, sometimes thousands of genes involved. And the individual contribution of each of these genes for these polygenic phenotypes is very, very small. So you would have to change 10, 20, 30 genes, and this becomes technically very, very difficult. So that's probably one of the reasons why the germline human enhancement will not take off, at least in the coming years.
Okay, so but you are talking about technical limitations, but you also talked and you said that in the majority of countries, genome editing, germline is not allowed. Mm-hmm. So, and I'm sure that's not only, of course, it's not a technical problem, it's an ethical issue, a religious issue. So I would ask João and Helena to also talk about that.
Sure, please.
Before I go to that, I would like to add a little bit more on the technical side, sure. Because I think that it's important that we have as much as possible clear image of what is at stake before we decide based on values, because we really need to know what is happening in the lab or in the hospital, if these things are going to the bedside as we hope to. And one thing that has not yet been mentioned by none of us is the on-target unrepaired events. So Vasco mentioned, rightly so, the off-target, the unintended changes in the genome, in the edited genome, elsewhere, apart from the target that we meant to modify, because it was the locus of interest related to the phenotype that we wish to change. But apart from that, and especially in embryos, we have the on-target unrepaired events, because the enzyme that the gene editing uses is a nuclease that introduces double strand breaks in DNA, in the target DNA. But the repair of these breaks is made not by any enzyme of the technology, but by the cell itself. And the cells have different strategies to repair DNA breaks. And we know that the embryos are particularly inefficient in repairing double-strand breaks. So we expect, and in fact, experimentally, this has been shown that edited embryos show a lot of on-target unrepaired lesions, breaks, and a number that in this recent summit on gene editing in London, they mentioned, one of the lecturers mentioned that at up to 40% of the cells of an embryo are not correctly edited. So it's, then in another study before, it was observed a chromosome loss, which is something. It's not just a point mutation, it's a loss of a whole chromosome. In the case, I think it was chromosome 6, because the locus of interest was in the chromosome 6, so it was a non-target, unrepaired lesion. So this is something that in terms of safety, we should, apart from indels, insertions, deletions, that in the lesion is repaired, the break is repaired, but with loss or gain of some genetic material. It's not a chromosome loss, but indels can have profound phenotypic consequences. But going then to a more ethical or bioethical stance, instead of just mentioning.
Can I just comment that what you have just said may also or not be an ethical problem. I mean, it's a technical problem, but then if you end up creating a lot of embryos in order to be able to obtain one that does not have that type of problems, you will have to end up discarding human embryos. Isn't that also, or may not that be an ethical?
Yes, but this is, as Vasco mentioned, just something that is already in the common practice of assisted reproduction technologies. The pre-implantation genetic diagnosis is exactly that. People, I even recently, one of my colleagues, female colleagues, they have produced 7 embryos or 9 embryos. They've implanted 2, and the rest, the remaining 7, were meant to be frozen for a second future attempt to, if the current 2 don't survive, later on people can thaw the frozen embryos and implant them. Unfortunately, the 7 died during the freezing procedure, so it was quite unfortunate, and this is her last opportunity, because it's already, she has attempted several times, and age doesn't stop, and but just going back to the discarding, this is regulated by law, even for research purposes. If the parents say so, these frozen embryos can be used for research purposes if they don't want them for donation. So our society has already come to terms in dealing with these extra embryos, but it has been a problem.
Yeah,
but which was, as many others, as you pointed out, in these 30 years of history, something that is very problematic at one moment. Later on, with the experience, the society changes, or the majority of society changes their minds, and people.
Yeah, I agree with you. I just asked about that because in my, sorry about speaking, my particular research, I work with induced pluripotent stem cells, and one of the main drive forces for Yamanaka to develop the technology was the problem associated with the manipulation of human embryos and the destruction of human embryos. That's why I know that is regulated in Portugal and outside of Portugal, but I don't know if today it's exactly like that, but there are still countries in Europe that, you can do research with human embryonic stem cells, but only if you import them from another country. I think Germany and Italy are these cases. So it's regulated, of course, but I think it's still, for some people, a matter of some debate. But I agree with you, it's totally regulated.
For these people that have some restrictions of using this technology, they are not obliged to.
No, of course not.
They are free not to embark in these lines of research. If I recall it well, embryonic stem cells are much better than induced pluripotent stem cells.
that would be another debate for this for the bioengineering.
But going back to the ethical of the human enhancement, this is the problem. Is apart from the technical complexity of multiplexing editing into numbers that are, with the current methods, inconceivable to change simultaneously hundreds or thousands of genes. Each one of those altering very little of the phenotype is the combination of the whole lot that gives rise to the phenotype, because these are clearly multigenic and multifactorial phenotypes. And this is something that we have not yet spoken about, which is the gene-environment interactions. The gene-gene interactions are already very complex, but if we add the environmental component of this equation, the things get even more complicated, because normal embryonic and fetal development is something that happens in a space and time that is physiological. The womb, and with some the mother's womb, and all the circumstances in which, in the time course of the events that lead to a normal newborn. Human or non-human, doesn't matter, the process is similar. So how can we reproduce in vitro these time and space events? It's very difficult. But before that is, are we going to agree on what is a desirable phenotype? Is something that we could call the human perfectibility. And I read an interesting article by a guy called Lagay from 2000, in which he puts this question: if parents would like us to design a baby with such and such features, what if later on in life this designed baby has a very unhappy life? And because the anticipation of the parents doesn't match with the reality that in which this baby is going to live 10, 15, 20 years later. Can we predict what are the best characteristics of a person in 20 years' time when it comes to the labor market, or to marry, or to the social life? And I think he is quite right in alerting us that what we think is good, or even worse, perfect, is maybe in some years' time it will be not interesting at all. And I couldn't help to recall the famous Rubens ladies that are very almost obese. They would like today, and this is 17th century, it was not in the prehistory, exactly in the prehistory, these Steatopygian Eves, these sculptures of the Paleolithic that have very, very wide hips women, because they would give rise to probably better children. I don't know what was in their minds, but the standard of perfectibility is something that is ever changing. And are we going to condemn, even if we could? Because my problem is technical, previous. I don't think we can. We can in terms of practicability, because of the gene-gene interactions and gene-environment interactions that we cannot reproduce in vitro. etc., etc. But even if we could, the result would be acceptable by the product of our design and by the social environment. I think this is something that we should keep in mind, trying to design babies or design cows or whatever.
I think Helena has a lot of things to say.
Well, thank you. I think that the first question that you were discussing, genetic preimplantation diagnosis, our law allows it, but only in serious situations. And we have a council, a national council on medically assisted procreation, and I belong to the council, and I am the only law professor. And so the professors of genetics, we have a list of diseases where it is already allowed, and every month we have new diseases, new mutations, new situations where the centers to medically assisted procreation ask the national council if they can do it or not. And it's quite interesting in the sense that they bring me the photos of the patients that have the diseases, and we are discussing it, should we allow it, should we not allow it. And the first conclusion that I have is that you are always finding new mutations linked with disease or disability. It is really we are very lucky with the genetic lottery that has allowed us to be here, because the probability of having trouble is quite high. But then we have situations where it is very easy. For instance, this month we had a disease in which the child would develop holes in the brain. It's a genetic mutation that is linked with creation of holes in the brain, quite easy.
Spongiform encephalopathy, maybe. Not
and we had a disease in which the child would be quite small, would have short stature. Yes, 1 meter and 50 centimeters. So the first one is quite easy, yes. No one deserves to be born with holes in the brain. The second one is, what is the problem of being 1 meter and 50? But the discussion is open, and it is a serious discussion, and every month we have it with the problems that you bring to us. And that stem cell research is a very good example of different laws that you have all over Europe, in the sense that at the national level, you have different legal systems, and you have legal systems where you can do whatever you want in stem cell research. United Kingdom, for example, you can even mix genetic material from human species with genetic material from other species. The limit is you cannot implant the embryos that you have created in the uterus of the woman. And then you have laws like Italy, where you can do nothing at all. Ireland, you can do nothing at all. Germany, you can do it with embryos that were not created with German cells. But you can import stem cells, and they are importing them from Israel.
From Israel, typically.
Yes. And this is linked with eugenics, and this is linked with the Nazi past, and this is linked with all the problems that the German legislator still has when he's trying to regulate this. So, first consequence, if you work in this field, you have biolaw tourism. And this means that you come to a person who works in biolaw, like me, and you ask, what is the best legal system for us to have our enterprise? And we will tell you, well, you should go to Dubai. And you go and you move. And that's terrible, because you're going to lose good research in your country. And at the same time, the results of your research, if they are not good, they are going to have an impact in the entire planet. You cannot stop a person who has received a xenograft, who has received an organ or a tissue that comes from a pig, from going to Portugal, to Spain, to England, you cannot stop the consequences of what you do within the national borders. So that is an interesting problem that if you work in this field, you will have to face. Another thing that is quite interesting is that the law that we have nowadays says that almost everything at international level, if you speak about UNESCO, if you speak about Council of Europe, says that almost everything that is linked with enhancement or germline therapy, gene therapy, is forbidden. But we have main principles, and the first principle is the right to inherit a genetic patrimony that has not been artificially modified. Another principle is the right to respect of your dignity and human rights, whatever your genetic constitution is. And your genetic constitution may be linked with disability or disease, as you know. And the third principle is that genetic information is very sensitive data in the sense that you have access to the information of your family, if we have access to your genetic information of your parents and of your children, and in the sense that we consider that genetic information was something that would not change during your lifetime. So these were the main principles that we had in international law for the last 20 years. But, and now I'm going to surprise you, we have this conservative approach to human dignity that says no to everything that alters the human species. And now we have a new approach to human dignity, a transhumanist approach to human dignity.
I'm not surprised at all, yeah.
And so what we are saying is you are creating embryos in different ways. We in law do not know what a human embryo is nowadays, because you could create it through fertilization, but you can create it through somatic cell nuclear transfer, but you can create it through induced pluripotent stem cells, or you can create it using stem cells, and then altering them and developing them so that they will be sperm or eggs, or you can induce an egg to work as an embryo, and then you have the European Court of Justice trying to define what a human embryo is for patent purposes. And then you have definitions in which we are beginning to be, you read it and you think, well, the court has gone absolutely crazy because they are saying, well, an embryo is an embryo that results from an egg that has been fertilized with a sperm. Well, but an egg is also an embryo that has result from a somatic cell nuclear transfer. And then it says, or any technique that will allow us to have in the end a baby. So whatever you do, we are going to try to regulate it. And we have used as well with the combined use of nanotechnology with biotechnology with informatics and informatic techniques that allow us to alter the human species in the sense that we have nanoproteins within our organisms. We have the use of stem cell tissues so the problem that we have nowadays is that with epigenetics, the idea that we had the same genetic patrimony from the moment of fertilization until death has disappeared. We now know, or you have told us, that different environments will allow us to have a different phenotype with the same genotype, and that the difference may be very serious.
It's very
serious.
Serious.
Yes. And that is why many times you see children that have very good education, very good health systems, very good nutrition, and that have lots of sports, being much more sometimes beautiful, sometimes healthy, than children that do not have the good conditions for their genome in the beginning. And you also have told us that you can change everything. And so what we are asking nowadays is, before we were the Australopithecus, and we were not happy, and we became the Neanderthal, and we became the Homo sapiens, and now we are the Homo sapiens sapiens. So is this enough? Should we say that our human dignity is linked with our biology? Why should we have a bioconservative approach to human dignity? Why are we not going to consider that we can be better, that you can create a species that will live longer, will have a better health, that will suffer less from disease, that will have good life quality in the last 20 years, that will be from 100 to 120. That in the end, when biology no longer can keep us alive, we will be able to put our software, our personality inside a pen and put it in a machine. So, what is a human legal subject? What is a human species? Should we move forward? And here you have two main approaches. One is the transhumanist approach that says, well, biology is still important. We want to go on as humans. We like being human animals. we are not going to consider, as we consider nowadays, our genetic constitution as human patrimony. And that is also a way of stopping your research, because another principle that we have is that we should protect as human patrimony our specific DNA, and that leads that everything that alters our DNA should be forbidden, having in mind the rights of the future generations that did not give their informed consent to be born with genes or with a genetic constitution that you have chosen. Mm-hmm. But we can say, well, we like our DNA, we like being humans. We will accept that you will enhance it a little bit more. We would like to be higher. We would like to have a better memory. We would like to be more resistant to work. We would like to have slaves. Because then you will have the relations between the people who have been enhanced and the normals, all of us. What is going to happen? And we will accept that. But then you have another part of the people who defend transhumanism that say, no, human biology is not enough. We want posthumanism. And this means that we are going to leave our biological constitution. We are going to merge with the machines. And we are going to become something different. Absolutely different. Cyborgs. And then we have another legal problem. What is the legal status that we are going to give to robots? And you already have Saudi Arabia saying that Robo Sophia from Hanson Robots, that is something really astonishing. I love Dolly. I love the sheep. I found it really something astonishing, and now I like Robo Sophia. And that say that Robo Sophia, even though she is able to learn, so she is not only a computer where you put the software and only does what we are asking her to do, but she already is able to do what babies do. She has the ability to learn. What are the legal status, or what is the legal status that we are going to give to her? And she already has legal personality. She's a legal citizen in Saudi Arabia. And the European Parliament is discussing the problem of the legal personality of intelligent machines. And then you have another problem. What will be our relation with the enhanced people that already are being part of our society? The treatments that you're already doing with stem cells, the treatments that you're already doing with nanotechnology, the treatments that you're already doing with informatics, the treatments that you're already doing with drugs. If you take a pill every day, you can be a very nice person. You can control your personality. You do not have to have your own personality with drugs, with pharmacology. Mm-hmm. You can have a new personality, a new, very nice personality. And we are doing it nowadays. The interventions that we already are doing are enhancement interventions in medicine. And the issue is, what will we be in 50 years' time? A society wants what you are doing. You are giving us better life quality. Of course, this is only for the rich people like us. It's not for Africa. In Africa, you will go on dying with malaria and cholera and so on. It's not our problem. Our problem here is having a very rich approach to healthcare. So, what are we going to do with the other animals that have the same DNA, as almost the same DNA as us, because we are big apes still? Mm-hmm. We do not think very much about it. It's not nice, but we are big apes, and the other big apes have 98% of their genome next to us. Should they be legal subjects as well? Shall we have two types of humans, the enhanced humans and the rest? What types of relations are we going to have with them? Will it be exactly the same as it was when we arrived to Africa and we had slavery? Mm-hmm. We will be the slave, will they be our masters in the sense that they will be better than us from a genetic point of view? And what is the legal status that we are going to give to the humans that will not have human biology? And I find this really interesting. And I think that law is changing very slowly, but we are opening the door to it in the sense that we have first, the Catholic Church and the conservative approach to biology are losing a little bit of space, namely in Council of Europe and the United Nations. And secondly, because we have to rethink if being human sapiens sapiens is enough. I don't know what you, the next generation, think about it, but would you like to be different and better?
I think Russia wants to.
Okay, no, that was surprising. I have to say. Yeah. So Elena took us to the year 2500. Perhaps our legal status by then won't be a problem because we'll be ruled by AI machines and we'll basically be slaves. So don't worry about our legal status. We'll be completely controlled by machines. No, just kidding. But now coming back to our age, I think it's very difficult to adopt a reasonable position when discussing these issues, because very easily we become hypocritical. It's much easier to be dogmatic, to defend, say, a maximalist approach, saying that everything is allowed. Let's be transhumanists or post-humanists and change our genome and fuse ourselves with machines, or be dogmatic at the other extreme and be bioconservative. As for instance, there are two people that you may be interested in reading if you are interested about these issues. One is Francis Fukuyama.
Yeah.
He wrote a book many years ago called Our Post-Human Future. I think it's translated in Portuguese. And the other is Jürgen Habermas. But go for Francis Fukuyama first, because Habermas is really hard to read. But he wrote a very small book on our human nature. And these two scholars, they are what we call bioconservatives. So they are clearly against any type of human enhancement based on genetic manipulation. And their main argument is that, and this now becomes a bit more philosophical than what we have been so far. Their main argument is that if we start changing our genetic structure, we are basically denying and corrupting our human dignity, and that liberty and our moral principles, they depend on keeping the genetic information as untouched as possible. Which is valid to a certain extent, but at the same time, it seems to almost sacralize our genetic information, as if by changing it, we are doing something that is forbidden by the gods. And it's not really clear why people that are born not being necessarily sick, but underprivileged out of this genetic lottery. For instance, 1 meter and 50 centimeters. This person is not sick in any way, but it clearly has some sort of disadvantage. It may not be tragic. We know very, very successful people that were not that tall. For instance, one of the most famous humorists these days is Joana Marques, right? She does the extremamente desagradável, and I think she's probably around 150, and it didn't prevent her from becoming very successful and famous. But still, there are a number of phenotypes which are at the borderline between what people would consider disease and what people would not consider disease. And this gray area is going to be a problem in the future. So again, it's very hard to try to be reasonable about this issue and find a middle ground. Even our society currently, I mean, we are living with a number of contradictions. For instance, we say that we do not try to interfere with the genetic composition of a person, but at the same time, we allow couples to abort embryos with Down syndrome. And we know that Down syndrome children and adults can live very happy lives if they are loved, right, and protected. but we don't like to think about this because these are really controversial and uncomfortable issues to deal with. So again, it's really hard to adopt a reasonable position in which we try to see everyone's point of view. My personal view on this is that human enhancement attempts are going to occur inevitably in the future, perhaps not in our lifetimes, but maybe in yours, okay? Maybe just within 50 years as we start seeing this type of approaches. And probably it's not going to happen in Europe, and it's not going to happen in the United States, but it will happen in Israel or in Singapore or in Korea or most likely in China. And as Elena was saying, it's really very hard to regulate these matters, it will only work if it's regulated at the global level. Otherwise, you create genetic tourism, right? You go to China and you start having babies made in China, right? And it's not only a pun; it could actually be a rigorous description of what we'll be seeing in the coming years. But at the same time, I think João is right, meaning there are so many technical problems that this human enhancement temptation raises, namely the fact that most of the appealing phenotypes are polygenic, so it's technically very difficult to change. And it's not only because many genes contribute to a given phenotype, it's also because a single gene can contribute to many phenotypes. So genes are pleiotropic. What this word means is that we may think that by changing gene A, we are increasing our memory capacities, but at the same time, we may be increasing our chances of developing a muscular disease. And there are actually examples of this possibility. So it's not only the polygenic nature of the phenotype, it's also the pleiotropism of the gene and the gene-environment interaction that João was telling us about. So we can't really always predict what's going to be the outcome, especially if we are dealing with complex phenotypes. And I can speak not only as a biologist, but also as being involved about 7 years ago in an experiment. I took a very minor role in it. My wife did most of the work. And we had twin daughters, and they are identical twins. So they should be identical, but they are clearly not identical in terms of personality. So genetics doesn't rule everything, and people should not sacralize genetics, which can be used as an argument for and against human enhancement. It's pretty much as you like it to use it. Okay.
I think what you were saying is related to the single question that we have from the audience, which is related to this question of what will hinder this genome editing from successfully happening. Are the technical issues, the technical limitations, or are the laws? Because, I mean, law is trying, as you said, Elena, to keep up the pace, but I think it's different. So what will you think it will be the main limiting step? Of course, in terms of law, as you said, you can always go eventually to China or to another country, and the law is different. Technical issues are probably more difficult to overcome. I don't know. What do you think about this? It's a question from the audience. João, do you want to start?
Yes, but again, before I go to that, this geographic relativism of what can or cannot be done. Surrogate motherhood, for instance, is a good example, because in Portugal we are still struggling with details of the law. It's not yet applicable, although we have a law of surrogate motherhood. Gestação de substituição is the Portuguese name for that. And when it will come, when it will be fully legalized, it will only be, it will be used only when the beneficiary woman doesn't have a womb. or a similar radical condition. Whereas, for instance, in Ukraine, you have a very open law, and they are providing surrogate motherhood for male couples, male-only couples, and for other reasons that are not allowed that will not be allowed in Portugal with the current law.
Issue in the beginning of the war because
Yes, they had a lot of babies that nobody would reclaim because they were afraid of going back to Ukraine to pick up the babies. So it was very apparent at that time. We didn't know, but then I went to the website of this firm, Ukrainian firm, and they don't even speak of therapeutic or medical reasons. Is mostly social reasons that they provide the service for.
You want? You buy?
You buy it. In
You
Dubai?
deliver? No, in Ukraine.
In Ukraine. Okay. So, but the question that you put is...
it's what will limit
okay.
editing for technicalities or law?
Well, I think that there are still a lot of technical hurdles to be overcome in terms of long-term safety and efficacy of the procedures, even in somatic gene editing, and also economic problems of equity of access, because the current methods or technologies, which are ex vivo methods in which you take cells from the person to be treated, you manipulate them in the lab, and you transplant it into the patient. It's very expensive and requires a sophisticated infrastructure. Whereas still being in an earlier stage of maturity as technology, in vivo editing, in which you have the drug, the editing machinery, and the guide RNA, the different pieces of the technology, and you inject it in the patient, so it's much easier. You can import, and then for instance, lower income countries where many of these single gene diseases, or the ones that have been more successfully treated already by gene editing, somatic gene editing, most of the patients live in lower income countries, for instance, in sub-Saharan Africa. It will be easier to apply this in vivo gene editing, although it is still as I told you, in an earlier stage of maturity as technology. So I think we still have a lot of technical and economical hurdles before it can be generally offered to the patients. But of course, in the same time, as in the background, we should be thinking of the ethical questions. And the guy that I mentioned earlier, that was saying that the couple that chooses a designer baby later on can be very, very sad because the baby finally doesn't match very well with the environment, the future environment. He was saying something very, very, very nice. He says, Homo sapiens should take its time in deciding whether the better gift to the future lies in manipulating the genome or letting nature take its course for a while longer while we watch and learn. Mm-hmm. Watch and learn. And this is why it's very important, as Vasco was saying earlier, to look at the natural mechanisms of gene editing, like in the immune system, or in bacteria or different models that we have, and try to understand how they work and what are the side effects that even in nature are operating, of course.
Now that we are reaching the end, I have here some interesting questions.
Margarida, may I just say very
Go
quickly?
ahead.
So going back to your question on what would be determining how these matters will evolve, if it's science or if it's law, I think it will be mainly politics, which of course will reflect also the status of the science and of the law, but politics in the sense that societies are becoming more individualistic, right? Not in the wrong connection of the word with becoming more selfish, which that may also be true, but individualistic in the sense that we will tend to respect even more in the future the rights of the individual, right, and the rights of the family. So if a given couple wants to do something which may benefit the progeny, and assuming that all these problems that João was referring to will eventually be solved, and that there will be no technical hurdle to surmount, and that there is no risk, and that indeed by changing gene A, C, and D, the child will for sure will have a much healthier and happy life. It will be very hard for society to forbid this couple to do it. I think the only force that can go against it is if it's not something that will be allowed for many people to do because of the price. If it's something extremely pricey, then there will be a strong opposition against it. And in democracies, probably that majority will prevent these technologies from becoming a reality. So that's one of the major resistance that I see preventing these human enhancements to be a reality. I mean, assuming that the technical problems are solved, assuming that people are no longer as bioconservative as Habermas and Fukuyama, and that they don't really sacralize genetics as some do these days. Issues related to equity, to see how is it possible to make these technologies available to everyone. I think those will be the central points of tension.
That is actually fully related with the question that I have in here. Who should take the responsibility for questions such as, is the Homo sapiens sapiens enough? Is a referendum, talking about politics. Elena, do you want to comment on this?
Yes, but if you allow me, I would suggest you to read. We are reading the same book. Nick Bostrom. Is really very interesting as well. And it's related with the question. It's not possible to solve this at a global level in the sense that you have many preconceptions at the national level, and there is no possible consensus. For instance, stem cell research, embryonic stem cell research. We have been discussing for the last 20 years in Council of Europe a protocol to the VIII Convention, that is our main legal text that we have in this field of biolaw in Europe, on the possibility of research in embryos. And the only thing that we were able to accept in Europe, thought as Europe from Lisbon to Russia, was that you should not create embryos for research purposes, and when embryo research is allowed, the national legal system should give adequate protection to the human embryo. As you can easily see, adequate is an open concept, and so you have situations or legal systems where you can do everything. You have legal systems like the Spanish legal system, where you're going to make a distinction between pre-embryo and embryo. So pre-embryo until the 14th day, where you no longer can have twins, and then embryo. You can do almost everything with a pre-embryo and nothing with an embryo, and so on. All over Europe, you have different solutions, and this tells us that from a global point of view, and this would mean United Nations, all that you can have are general principles. And that is the possibility now in the 21st century. And another thing that you refer that is very interesting is the problem of democracy, in the sense that when you speak about diseases like nanism, you have people that suffer from genetically determined nanism that say we are very happy to be small. We are a new minority. The problem is you. Small people, they tend to marry with people who suffer from the same disease. They have small houses, small furniture, and they say, we want genetic preimplantation diagnosis to have a child that will be small as well. What are we going to do with the master? But if you move to the deaf community, you'll find exactly the same thing. They will say, we are not disabled. We are not people with a disease. We are simply different. And we want to have children who suffer from deafness as well. It's not good having noise in your head. We are a cultural minority. We have our language, sign language. We have our culture. Respect us. If we move to Down syndrome, we have people that say suffering from Down syndrome is being genetically different from the majority of the population. What is the problem? Respect us. Allow us to be born as people who are genetically different. And then you have another part of the society saying we do not want to have a child with Down syndrome because we are in a highly competitive society that does not have space for everyone who is normal. So if you have a child that already has something that is looked as a non-advantage, as a disadvantage, that child will not have her right to an open future respected. And that right means that you should give the best genetic startup to your children that you can afford to give through genetics. And so this is also a problem of discrimination law, anti-discrimination law, and the relations that you have between the majority and the minorities. And what is really the meaning of what are we humans? What does it mean to be human? And then when you move to who is going to stop this? No one. History has shown us that what is technically possible will be done. So, and you are very good in what you do. You like very much what you do. When you are doing it, many times you're not thinking about bioethical issues. And I can give you an example, human reproductive cloning. You have a very interesting decision of the European Court of Human Rights on the Raelian religious sect, it's not really a religion, that say many years ago, more than 10 years ago, that they have already produced the first human clone, Eve. But they will not tell us where, because it's forbidden in many places. So probably where they have created Eve, and as you know very well, cloning with one mammal means that cloning is possible with another mammal, and we are mammals. Another bad. So it's technically possible. So probably it's being done. And we will not even notice, since if twins are different, a clone that will be born with a 20-year span will not be a child. Any clone, a clone of Mafalda, nowadays a small baby, we would not recognize the clone as being a clone. So how can we stop this? We cannot stop this. It's not stop. What we can do is try at a national level and within the international organizations that we belong to, in Portugal, European Union, Council of Europe, United Nations, we can try to preserve the most essential principles around values and concepts of justice that are different from country to country and even within the same countries, and see what happens. And as we have wars, and as we are think about the situation that we are living now, with the possibility in 1-2 months of having rockets in Lisbon. Think about what we are doing with the climate and the possibility of destroying our species without having water or something else. So the problem may be solved with all the other problems that we have around. But from a technical point of view, I think it will be done. From a bioethical point of view, probably it will not be stopped because people are liking it. Think about all that we are doing in pre-implantation diagnosis, in prenatal diagnosis. Look around you. Almost everyone the children that are not born with diseases or disabilities within families that suffer from these genetic determinants. So the society is liking it. It's a silent revolution. And as you said very well, it's not a decision that the state imposes. What happens is that each couple decides. So you have Huntington, you have another monogenic disease. You go to you go, and you ask, what can we do? And they will tell you, well, you can have in vitro fertilization and select the embryo that does not have the mutation, and you will do it. And the disease will disappear in your family. So it's something that, and you have also the genetic market, and that is the genetic supermarket. And that is how it is being discussed nowadays in the United States legal systems. That means that if you have an offer of genetic services, someone will buy them.
Yeah.
And people are happily buying them. And if you have enough money, your children will be more beautiful, your children will be more healthy, you will not have children with disabilities, you will have the possibility of choosing the best genes for your child, and you can select the donor. I don't know if you know, but half of the Portuguese men, when you're speaking about sperm donation, are rejected. They do not have enough quality in their sperm. So if you have a child with sperm donation, you will have better genes for your child than if you have a child with your boyfriend that has not been genetically controlled.
Não deve ser por razões genéticas que eles não fazem estudos, não?
Fazem, os nossos centros de...
No, the quality of the sperm is essentially the number of,
Yes.
of sperm cells and the mobility of
Yes.
the sperm cells, and only a few genes are, if
Yes.
are screened for, like cystic fibrosis and a few other. So we don't profile extensively the sperm, but of course there are quality issues, mostly of environmental cause, because we live in a polluted environment. We eat things that have residues of pesticides and other endocrine disruptive molecules, and in consequence our spermatogenesis is badly influenced by environmental issues, and this is probably why so many donors are rejected, because of not really for strictly genetic reasons, because I don't think that we are in the position to do a full genetic screening of the clinical genome. Includes about 5,000 genes, at least 5,000 of our 20,000 more or less genes of our genome have been associated to a disease.
Yes.
And certainly they are not doing as a routine screening of 5,000 genes that could be the reason to exclude a donor, a sperm donor. But of course, the environmental situation is not good.
Yes, what we're discussing nowadays is, should you, traditionally we had children with sexual intercourse. In the future, probably your generation, if you want the best for your child, it would not be rational to have children through sexual intercourse, in the sense that you should have the best sperm and the best eggs, and you should try to detect the diseases that you could avoid. And if you have a sperm donor, at least it will have some genetic quality screening, and we are quite careful in it, in the sense that this is a very expensive service. If you are young, nowadays one of the problems that we have in infertility is that women are having children later.
Yeah.
The best
age is very important.
It's a very serious problem.
Yeah.
You should be having children now if you wanted to have really good children. So you should preserve your eggs. You should freeze your eggs. And in 10 years' time, and we are accepting the freezing of eggs for social purposes. So you should freeze your eggs, you should go on with your degree, you should have a very good job, and then finally you should ask for some sperm from the bank, the best quality possible, and you should have a child with your frozen eggs.
See, very, very practical advice. Besides politics and philosophy, now you know what to do.
It's what is being done, because this is our society nowadays. We are discussing what is the age limit for a woman to have access to medically assisted procreation, because women are having children when they are 38, 40. And you have a different age limit if you go to the public system or to the private system. And you are discussing if you cannot have a child when you're 60 with eggs of a lady who is 22. And you are discussing if you should select the sex of your child, because another discussion that we have here is sex selection. You can easily select the sex of your child. And our law in Council of Europe says that you can only do it if you have a genetic serious inherited disease.
An excellent.
But in China, in India, they are not doing it for these proposals. And they already have more women than men. And this will bring wars, because when men do not have women to marry, they are going to try to find them in the neighbor countries. So as you see, you are going to decide the future of humankind, and probably you will do it well.
Okay.
Everything has happened well until this moment. There is no reason to
No
think
doubt.
it will be well for the future.
I think we have to finish now. I had here some more questions in which the students demonstrate somehow their concerns about, okay, if we can do genome editing for human enhancement, what will happen to people that do not have access to this? This is a very important ethical issue, because we are talking about expensive technologies. And also a very interesting question about what will happen to humans' natural survival instinct if you are able to solve everything, basically, with technical approaches. So probably I would ask each one of you to make a wrapping up final comment. And if you want to approach some of these issues, please go ahead, and then we would finish. Okay? Do you want to start, Vasco, and then João and Helene? Wrapping up.
Okay, well, I would just say that if you are interested about these matters, there is an extensive literature that you could start reading, and I will be more than happy to send you what I think are the most relevant authors and texts that you should read. So just email me, and I'll send you my favorite list.
Okay, that's a good advice, because we always need to have robust information to be able to have an opinion about everything, and mainly about these issues. João?
Well, I don't think I have much to do beyond what I have just said, but to me, the bottom line still is that gene editing is a promising technology to be used for the moment in conditions which have a simple genetics, ideally monogenic conditions, and in a somatic tissue. To me, this is really the baseline. Even though with further safety, long-term safety evaluation.
Yes, I think that human dignity is the main value that we'll have to respect when deciding if we are going to use or not what you are doing in your lives, and that inequality has a new face in the sense that now we have genism, a new type of discrimination, genetic discrimination. And genetic discrimination is very serious in the sense that we had natural genetic discrimination. That was the first lottery that we had, no one has chosen the genetic discrimination that we have. And now we'll have genetic discrimination that will be something that will result of what is going to be done with us during our lifetime. And this means that we'll have new types of inequality. The very rich will be enhanced, the very rich will have a better genome, the very rich will be happier, will live longer, will have life quality. The other ones will be subhumans. The normal will be not normal, we'll have a new normal. And we'll see what relations we'll have between them, but the relations will probably be bad in the sense that some will be naturally better. And we already have some that are naturally better.
Better off.
Better off as well. But you'll be able, before you were not able to buy it.
Yeah.
And now you are beginning to be able to buy it. So only the rich one will have access to it. The rest of the humanity is what we have nowadays. We have a medicine for the northern part of the planet and a medicine for the southern part of the planet. We have the northern part of the planet living well, the southern part of the planet with $1 per day. It will be worse than it is nowadays for the other part of the planet.
It's a new reality.
Yes.
Okay, so I think we have to end now, and let's give a big applause to this great panel that was here today.
Muito bom. Fantástico. Depois não davam as perguntas do computador.
Okay, just before closing the session, I just wanted to announce the winners of today's session, Francisco Fragoso and Inês Carvalho. If you are here, you can come and run your prize. And once again, I would like to thank you all for being here, to Professor Matheus and to our speakers. Thank you so much for this interesting discussion. Thank you.
Thank you.
Muito bem. Foi muito giro. Sim.